So how many of you were stressed or frustrated just now when the unzip undone thing wasn't working? Okay, good. That's how you're supposed to feel. Yes. I'm trying to, yeah, but anyway, I apologize for that. Yeah, but it's going to be fine. Yeah, it was just the file. The file got. I mean, it was working for me. It was working for some of us, and then we were just wondering why isn't it working for everyone? But at least you feel that frustration, so it's good. All right, so now we will go to the next level of not so much frustration, I hope. And then, so yeah, so here in this module, module 3, so every module is supposed to be a follow-up, right, of the previous module. So the first part you're talking about, Savannah brought you through cleaning up QC, how to exactly look at your data, right? So this is your raw, fresh data out. You're looking at accounts, you're looking at your features, genes, and you're trying to decide, okay, how much do I clean it up? And then you're going through that first process of normalization. And then after that, right, you really want to get on with your analysis. So you want to, let's say, start to run methods that will help you understand biology, right? So one of the tools that you will commonly use is really trying to build your spatial model. I mean, that is why everyone wants to do spatial, what do you call it, analysis, is because you want to see the context of it. So these are some of the learning objectives. So it's really, I'm hoping that you will learn to think critically about spatial biology and analysis, and also to develop an intuition on the factors that affect or influence spatial analysis, and some foundational skills in spatial analysis. And don't have to, I think everyone will develop or will form their opinion and compendium of favorite spatial tools. There's no right or wrong answer. It really is understanding what is going on in the background, all right? And you all know your projects better than anybody else. So yeah. Okay, I think they should have gone before the learning objectives, but never mind. Okay, so why do we want to cluster? So these are all the steps that I'm going to go through. And then based on all the questions that you've been asking and posted on Slack, what I'm going to do is at the end of this lecture, before we start practical, I've chosen a select few of my favorite papers, which I'm going to review through using the materials and methods and the concepts, because I thought I'll share with you how I went through the process of choosing the methods for my spatial analysis. It's like a sort of sharing my own experience rather than just all theory. So why do we want to cluster? Okay, so this is work that I've been doing, but on the Xenium, right? So why do we want to cluster? So I work on glioblastoma, and glioblastoma is very, very heterogeneous, right? So you have cell types that are very mixed, very intermixed, very distributed. But the thing is, when you look at histology and when you look at broad domains, you will find patterns. So this is just an example of some of the patterns that I see. So on the first, what do you call it, image here, that's actually a sample that has been labeled with cell type label and specifically transferred from GBMap, which is a single cell data set, right? This is something that we all used to, that we've done during single cell analysis. Now, this is a Banksy clustered domain plot here. So these are all your spatial clusters, all right? And then you start to see something that is broader. So you have all these clusters that occupy a region. So that's what you're supposed to see, or you think you should see. And then what is it that you want to get from spatial clusters? You want to get that understanding that if I can find a domain, or I'm hoping to find a domain, that domain will have a certain function, or it will have a certain cell type that is aggregated, or cell types that are aggregated. So in this example, and this is something that I picked and chose for this presentation, it doesn't always work that way. So in this case, what you do find is that you have two different spatial domains, and they are enriched by different cell state signatures, right? This is like the best case scenario, but it doesn't always work that way, because even though I'm not able to share some of these images that analysis that I've done, but I can definitely tell you that not all disease tissues actually have spatial domains. And it also depends on the region in which you are actually cutting or slicing or taking your tissues from. So when it comes to spatial analysis, whether you find domain or a non-domain, just non-spatial clustering, it really depends on where you're interrogating it from. So it's not as simple as just passing it through an algorithm. So why we have to critically evaluate why do we want to do spatial. So when it comes to my project, when I started it, I had gone through some experience with sitting with a pathologist who would annotate these regions, right, for me. And so I was lucky enough to have that information that I could use and convince myself that spatial clustering could actually be an avenue to explore, that I would actually get an answer out of it. But sometimes you don't need, a computational method to evaluate or to come up with spatial clusters. So in this paper, what the authors have defined, specifically what they want to understand is the question, right? So the question is they want to understand the gradient of gene expression patterns between, let's say, different histologically defined domains. So this was manually annotated. So you don't need to have an unsupervised spatial clustering. You can have your own clusters that are predefined by you, annotated by you, and you can split your gene expression through that as well. All right. So what it means to actually have spatial clusters, to do spatial clustering, is really to understand domain structure, broad collectives of cells. And that should be driven by knowledge of your tissue. All right. And if you don't have knowledge of your tissue, I think you should start with the simplest method. All right. You should look at your cell type profile, see how it looks compares with your single cell data. Look at your H&E staining and see if there's anything obvious. We kind of underestimate the amount of information that we actually can see through our own eyes when we just load an image, right? So use all of this. And I would also start with just simple non-spatial clustering, all right? Instead of just jumping into spatial clustering, because there are many different methods. And if there is domain structure, most likely your non-spatial clustering will pick it up. It just won't be so clean, all right? And okay, so spatial means we want to understand networks, interactions, communication, and patterns that are beyond linear, right? So you have, for instance, every single spatial clustering method, they benchmark against the mouse brain. How many of you agree with that benchmarking standard? No one? Really? Really? No one? Oh, okay. Yes? No? Okay, I'm going to take it's a no, right? Okay, so the thing is, the mouse brain or the human brain, right, is very well structured. And okay, Javaria is looking at me, so is the liver. All right. Huh? No, I'm talking about structure. So the liver is also, yes, yes. Yeah. So, yeah. So most of you are looking, I think half of you will be working on brain. So it's very well structured, all right? So you have your layers, right? But the thing is, in some of these tissues, the layers have a very strictly defined cell type proportion, cell types that are happening. So it's not so difficult to benchmark against something that is already beautifully structured, right? But what about if we are actually dealing with something as complex as disease? How are you going to know if your spatial clusters are picking up the right domains? Many of these spatial methods, if you really think about it, on one hand, you can have it guided by a pathologist or even guided by your own experience, right, on what you know of your own tissues, and that's good as well. But on the other hand, the reason why I emphasize on looking at your materials and methods, looking at the results, all right, really evaluating what the authors are trying to do is that many of these are actually smoothing algorithms. They are smoothing, all right, they're trying to find common patterns and they're trying to group these common patterns to you. So how do we know that in a diseased or in a treated state, that domain that I'm seeing is actually factual? All right, so these are all the things that we need to consider when we want to look at patterns in tissue. But that doesn't mean that it's super difficult, all right? Just that don't be too some of you asked this question, what is the best method? I think the best method is usually the easiest method. Try the easiest method because all these algorithms, they have flaws. But if you look at all benchmarking standards, they kind of still pick up patterns in one way or another. So you evaluate using the simplest method first, all right? You get over that first threshold, all right? Once you convince yourself that there is something there, and then you can fine tune and refine, and then you can try to explore something that's more complex. If you need a graph neural network, do you need more training, and so on and so forth. So going along this line of argument, but the other thing that we really need to consider, and something that I guess when I started doing spatial clustering, it looked so nice, right? So everything I wanted to do, spatial clustering. So I tried SPA GCN. I really liked Banksy, but I came across a couple of tissues where I do not require spatial clustering. I could have just done non-spatial clustering and it would have given me all the domains because the cells are so well organized. I don't need anything more than that. Right? And it also really simplifies your research as well and to kind of move you along. Yeah. So spatial is the understanding of collective mechanisms or identifying domains. All right. So the start with the simplest model. Okay. So the next thing that I wanted to explore was non-spatial versus spatial. So this is the part where I wanted to talk about. So I use Banksy in all of my teaching sessions, and I use it for my own work as well. I've tried a couple of others, but not too much, because I don't think I need to try 10,000 different methods as long as I know what I'm asking and I can see the features. And I honestly don't think we have time to do it, to go through all. But the thing that I wanted to point out, right, if you look at this image, that's non-spatial and that is spatial. What spatial is doing simply, despite how complex the algorithm is, all right, or how complicated you think it might be, it is simply smoothing regions that are already identified by non-spatial. So this is what I mean by you can already see the patterns when you do your cell type labeling. You will already see those patterns when you do your standard clustering. Can be any kind of clustering. Even the simplest methods will pick up these collective organizational patterns. I've done something that is just on phase contrast images with no single cell, matrix at all, and I've seen spatial patterns, right? So if it's there, you will see it, all right? Which slide am I in? This one. I think I put too many slides here. Okay, so now let's talk about how spatial works, okay? So this is the part where I think parameter fine-tuning and the actual algorithm that you want to use actually comes into play. So Banksy is, I won't say it is the best. I don't think there's such a thing called a best method, but it's just that it's very logical. All right. So logically, when you do spatial clustering, what is it that you want, right? So you do want to take the gene expression of your cell, right? So every cell has got n number of features and they all have a certain expression level, right? But that is something that you do even in single cell, right? So that's the baseline. You need that. You cannot do without features. But the next additional layer is that you have an additional matrix that's going to give information about the distances between these cells, all right? How they're organized, whether they're close, whether they're far apart, or whether they're clustered together. So Banksy has got 3 different matrices, all right? And I'll talk about this more. So it's a very clever, intuitive way, and it's logical. So they have one layer, all right, of your matrix where they look at just the gene expression of each cell. And then the second matrix is basically the average the average gene expression of the neighborhood of each cell. All right. And then the third layer, what do you think it is? Very often when we look at our tissues, all right, depending on the region, depending on the disease, what we find is that even the same cell types can have different cell states. They can have, what do you call it, a highly variable component of their genes, all right, that is completely different. So you can have the same cell type, but 2 different cell states, all right, undergoing 2 different transformations. So Banksy takes into that logic, and they borrow this idea from computer vision. They use a Gabor filter. So a Gabor filter basically is if you have light waves, all right, which are passing through, and then you apply a filter, usually it's a Gaussian filter, and then it kind of picks up, all right, certain peaks from it. So in this case, it's actually picking up the gradient of gene expression, all right, in a circular fashion. So it's very sensible, right? It's a very sensible way. But just because something is sensible, it doesn't mean that it will work in practice, right? So that is where your experience comes in. So that's how it works. But the thing is, I find that most of these algorithms, they work in the same way, right? So you don't have to spend too much of time, all right? Sometimes the logical way to do things is to really understand how well, I like it when in papers, authors explain their materials and methods in a very approachable way, in a very logical manner. When it's too complex and I cannot understand it, maybe it's me, this is my personal opinion, all right? I find it harder to trust the method and the process because I feel like I cannot troubleshoot it. Like, why would I want to do something that I cannot explain to myself, right? Yeah, so I think that's where you should start. And then the second level is, all right, it's also you need to go to the GitHub repo, make sure that the authors are people who actually update and keep their GitHub repo updated. I think this is logical, especially for those of us, all right, who are not spending so much of time into method development. We are applying, right? So we need to be logical in that as well. So for instance, COVID, there is this method called COVID. I really like the logic of it. It's from Dana Pierce's lab. And it was so well structured, beautifully written as well, but it was just impossible to process. Like my memory kept failing on me. My VMs kept shutting down. Like you don't want to go through that process. All right. So these are just logical things, nothing to do with spatial, but yeah, it's just the process. But just to let you know that don't get too frustrated if something you like doesn't work, because I really think that most of these algorithms, more or less, all right, they're going to give you good results, just that some will be slightly better than others, and it depends on your tissue. Okay, now this one. I know that thing about spatial, when we start to do things spatial, everything started to come into a mix, right? So then you have the question of segmentation. How is segmentation going to affect my spatial clusters? How is the bin size or my scale size going to affect what kind of kernel smoothing? Now, this is a problem I find. I think this is a problem I find when you're doing tissue microarrays. I have most of the time, my failures when it comes to spatial clustering and identifying domain have come when I use TMAs, tissue microarrays. But also, right, take note that I've been using tissue microarrays that are diseased, right? I'm not using normal tissue. I don't work with normal tissues. Most of my tissues are just GBM or some sort of cancer, some sort of disease. And when the size is small, when you only have a 1 mm TMA core, all right, the robustness in which the algorithm is able to smooth and identify domains is going to be pretty much a little impossible. All right. And I think I have an example of that soon. So before that, right, I just want to go through this step by step. So for segmentation, right? Okay, we're talking about Visium HD here. Visium HD is not single cell. It is single spot. It's subcellular spots, right? It is different. It is basically you have tissue that is over this grid, all right? And each grid is going to extract a set of counts, all right, from that region within that 2 micron by 2 micron. It's not single cell, all right? It's just, it can be part of a cell. It can be between boundaries between two cells, all right? Now, whether segmentation will improve your spatial clustering, I have avoided segmenting any Visium HD, all right? And truthfully, you don't need segmentation. You can see the domains, you can see your clusters, you can see those spatial patterns without segmentation. And it's probably the easier route to take before you segment. You only segment if you feel that there is value to it. So some of the issues that comes with segmentation, right? I know that sometimes the code, everything looks fine, but there are some who have experienced that the nature of the cell types that populate their tissues, right, actually results in a lot of bins that are dropped off after segmentation. Do you want that? What if you actually want to keep regions that are acellular as well? What if your primary purpose is not cell-cell communication or something that's granular, but you want to see trajectory of changes across a spatial domain, right? So ask yourself these questions and don't force something into your pipeline unless you really need it. And the thing is, right, after you've gone through this and you've seen the broad picture, you can always come back again and apply segmentation, right? There are tons of it out there, but get the broad picture and understand. And then once you see those spatial patterns, then you can ask yourself, is segmentation is going to give you any added value to your spatial clusters? But because the nature of spatial clusters is to identify broad domains, I don't think that would be the first step to jump into something so difficult as cell segmentation. Yeah, I spoke too much about that. Okay. Now let's move on. Okay. Interpretation of gene modules or differential expression. I don't know. I feel that differential expression is suffering a little when it comes to spatial clustering because the statistics is different, right? So for those of you who know statistics, and my statistics is not the best, it's not my best at all. I didn't like the topic when I was in school. But the thing is, we don't have the proper statistics developed for, I think, spatially aware differential analysis. Like we are still in the process of understanding, testing things out. So I cannot say for sure that when you get your spatial clusters, your standard differential analysis, all right, expression analysis will give you the enriched genes, all right, that are biologically reliable. A lot of collaborators that I've worked with, it seems to be that so there's a collaborator where I did just tissue microarray course, but I didn't do spatial clustering. I just did non-spatial. I did cell type labeling, and then I did gene expression, differential analysis, and it seemed to work fine. It enriched for all the right genes. But when I applied spatial clustering and I tried to do differential analysis, all the p-values were all off. It just didn't make sense. And the heat maps were all wrong, right? So I think the stats has to be applied properly as well. And the thing is, like, for instance, you also have to be careful. You really need to understand what is the output from your spatial clustering. So Banksy, right, the output is actually, you will find that your matrix has increased by times 3 because it appends the next to the average gene expression neighborhood and also the Gabor filtered gene expression gradient matrix to it. I have tried to perform gene expression analysis on all 3 because I thought maybe the third matrix would give me something interesting because it's based on the azimuthal Gabor filter, which looks at gradient of gene expression, but it didn't work. But it was really giving me garbage. The only thing that was making sense was actually the first matrix, the original gene expression matrix, the single cell matrix. But that could be because I was very naively applying, all right, the standard differential expression analysis from ScanPy, right? So maybe I should have done something. So these are things that you have to think about. But the thing is, sometimes differential analysis can be just between 2 regions. So in this paper, going back to this paper here again, what they did was they were really smart. I think they had a very clearly defined question because the author is a neurosurgeon. So I guess they have a clearly defined approach. So they selected just 2 regions, all right, and they did differential analysis and they concentrated their focus on understanding how the genes changed or how the genes varied across this. So just be a little, I guess, based on what I've gone through, just be a little careful when you do differential analysis, all right? Make sure you understand what your matrix output is before you venture into that. Yeah, so in this case, sometimes, right, I think all this goes into the fact that sometimes some of the spatial clustering methods, they are only interested in domain identification. So the output is of no use, perhaps, all right, for differential gene expression analysis. And I've been reading a lot of papers on Banksy. I have not found, and you can correct me if I'm wrong, I have not come across any paper that has applied differential gene expression on a Banksy matrix yet. All right, so it could just be the nature of so be careful to take all of this into account when you choose your spatial clustering. Yeah. Okay, so yes, I think I've already said this. Think critically of tissue type and tissue size. It really matters. Some tissues have patterns that do not necessitate spatial analysis. Yes, so the scale of pattern recognition, that's what I wanted to talk about. So why the size matters is because it's the scale of pattern recognition. So, there's this famous thing in geospatial analysis. I forgot the author. I don't remember names very well. But anyway, the thing was, if you map, right, the coastline, and as you zoom in and you map the coastline again, and you zoom in and map the coastline again, the length will get longer and longer and longer. That is because your scale of measurement is changing. All right. So in the same way, if your pattern that is there, present, it is just beyond the limits, all right, of the algorithm that you're trying to run, you will miss it. So it doesn't mean that it's not there. All right, it's just that is the limitation of your experiment. Okay, so these are some of the, so all the spatial clustering methods that you will come across. In fact, segmentation is the same thing. So for all kinds of segmentation, and we're not doing segmentation for this workshop. We did it for the Xenium. So you can go and look at the ISO 2024 workshop. But we're not going to do it here, but it's the same principle, right? So how you define boundaries and how you define domains. Domains are also kind of boundaries, right? So there are two ways to look at it. One of it is you can use a graph neural network. You can also train it, all right? You can also train it on, what do you call it, H&E, or you can purely just approach it from a statistical point of view, which is much simpler and it gives very, very reliable results. But most of these approaches fall under these two categories. It's either graph-based or statistics-based. But for segmentation, I have to agree that the deep learning methods are much better than the statistical methods. Okay, so yeah, so you see this one is from the Allen Brain Atlas. So you find that many of these benchmarking papers, they'll have all these nice, beautiful pictures of the mouse hippocampus, dentate gyrus. I mean, the structures are all well preserved, right? But you're not going to find this when you're looking at something that is diseased. Okay, so this one is another example. So this one is a nice example. So this is a GBM paper. So in this case, what they have actually identified is that you have GBM tissue and you can collectively divide them into two different types of patterns. One is organized and one is disorganized, right? So imagine now this is where the concept of scale comes in. If you have, so theoretically, all these algorithms will be able to pick up a domain that is heterogeneous, right? And it will just form one big cluster. Okay, this is a heterogeneous cluster, all right? And it belongs to one domain because the heterogeneity of the cell types, perhaps the cell types belong to only a certain subset of all cell types. And the nature of that disorganization, all right, means that when you smooth it, you find that the pattern is similar across this region. Okay, so it should be able to pick up even a disorganized structure. But now imagine you punch a tissue microarray core, 1 mm, all right? It is not broad enough to see the patterns because it doesn't have information where it is organized as well. All right, so it will not be able to compute. When you perform your principal component analysis and your Leiden clustering, it will not be able to see. All these methods, in a way, they depend on relative measurements, right? They look at your whole data set and then you're trying to understand and see what the variation is. If you have a small area and it's not going to give you that much of spatial pattern variance, you won't be able to pick it up. Yeah, so this is actually a good example of that. Okay, so I thought maybe right now, okay, let me see. See what I will share with you so I don't share the wrong things. And I have a lot of windows on my. Do you have any questions? Does anyone have any questions?
I had a question about with the differential expression in Banksy. I was wondering if the Banksy, I guess, quote unquote, corrected counts, I don't know if that's the right term. If those counts aren't really ideal for looking at differential expression, could you use, or would it be appropriate to use the labels generated by Banksy for the domains and then go back to the initial unsmooth counts and then start looking at differential expression between regions?
So you can, right? And the first matrix of Banksy is actually your normal generic counts matrix. So yeah, so I've done that, and that is the one that usually gives me the most reasonable. Yeah. So you can do that. Is that the only thing that, because I'm not a statistician, the only thing is that I always wonder if statistically that is significant, which is why you have to do a bit of validation after you pick up all the genes. Yeah.
Okay, thank you.
From Nazia, is there a gold standard threshold for the area of tissue?
Bigger the better. So long answer. It's a gold standard. I think it depends. Okay, so the difficult thing about spatial, right? And I went through this and I completely missed it. So we had a Visium HD experiment, and it was two sister sections. All right. And the two sister sections were just slightly off each other. That's it. Okay. It just happened to be that the second sister section had got more features, more domain features on the left side. So when I analyzed the first one, all right, I had less heterogeneity in spatial. So it's not really about the size. I think, to be very frank, the size of the Visium HD, all right, that imaging size is fine, is okay, really. You can capture many features, many domain features. I think what is actually more important is the sampling.
Hey, how to evaluate the accuracy or correctness of the identified clusters?
Well, there are a few, you can use like the classical, the adjusted RAND index. You can use normalized mutual information, homogeneity scores. Like if you go to, like computer vision has got a whole string of all of these heterogeneity, homogeneity scores, and you can evaluate how close it is, I guess, to the ground truth. But all of that requires some sort of ground truth. Yeah. I don't think there is an absolute measure to quantify this when it comes to something where you are discovering spatial clusters or domains.
Hey, what if the tumor biopsy is very tiny? How would that influence the clustering or analysis?
Yeah, I would suggest not to try like spatial clustering. No, no, really, really. Like try, like do it simple. Like try non-spatial, do your cell type labeling. Um, Understand where the tissue or the biopsy came from. And I think I think some of these, yeah, it's difficult, right? Because so the thing is when you can try to integrate. You can try to integrate these samples, but when the tissue size is too small, I have always refrained from not doing spatial clustering. I would rather go back to the HNE. I'm not sure if that is the answer that you're looking for, but yeah.
What is the benefit of using Visium HD and then running an algorithm like Banksy if it does not cluster at a single cell resolution? Would it be more efficient to use standard Visium for this type of approach?
So the thing is, right, we must detach spatial clustering from the resolution. Spatial clustering is there to identify broad patterns. So it will do well even if it's a 2 micron Visium HD or it's a 55 micron Visium. That doesn't matter. I think the problem that I've seen so far comes is really dependent on the region and the size of the tissue, all right, and really the nature of the tissue itself. But all spatial clustering, what is spatial clustering? It's not there to look at granular details. It's looking at your gene expression and how it changes across XYZ or XY, all right? And the general gene expression of each cell, and then it's trying to compute how cells or spots are collectively working together.
Thank you. Going back to the sampling issue, in the case of high heterogeneity and in practical terms like cost, what do you suggest for sampling best practices?
Oh no, I know, it's very expensive. Um, Ideal case. So, okay, let's remove the cost, right? we had the same problem in single cell, right? We used to talk about sampling as well. It's the same thing. So remove the cost. What would you do if this was a single cell or a bulk sequencing? It's the same thing. When bulk sequencing started, we had one one sample per patient, or per biological entity, right? And then we realized that, oh, that's not enough, and then we subsample from different regions, right? So it's the same thing for single cell. So we know that, when you take the I'm going back to GBM again because that's what I work on. So if you take the tissue from the core and you take it from the edge and you take it from let's say into the infiltrated region, all right, these three different samples are going to give you three different sites. So it's like n equals to 3 for one sample, three different sites. And then there is heterogeneity within, right? So you account for that. And then papers started to come out. All right, so we need to look at the HNE, and then we need to profile certain regions, and we need to maybe make multiple samples. So it's the same analogy. All right. So the thing is, if you think that your question is to ask, I am looking at some sort of a sequencing analysis, and it's a patient sample, and you want to find something that is targeted, that's something that is therapeutically relevant, all right, what would you do? You would actually sample, all right, based on surgical guidance. You'll take more than 2, 3 regions, right? And if you are looking at untreated, treated, you will follow the n equals to 3 rule. Yeah, so this is minusing off the cost, okay? Yeah, so ideal case, that's how you approach. Yeah, and that is how we have handled all experiments in the past as well. The logic is the same, it's just the platform has changed.
Hey, and I'm going to ask another question, but if anyone in here has questions, come on up. It's totally fine. How do you decide the right clustering resolution since the clusters look completely different on different resolutions? Clustree does not help here either.
Yeah, that's true. Clustering does not help. It's very frustrating. Yeah, to be very frank, right now, everything that I've been doing is just based on HNE. So for me, I have a HNE of my GBM samples, and I look at it. I look at Sankey plots. So I try out multiple resolutions. I look at Sankey plots. I look at how my cell type profiles change. I do a lot of cleanup. I remove like really low expressing cells. I do try to look out for resolutions where my clustering has become stable. Like if I realize that it's becoming unstable after a certain point, I kind of go back to the point. Sometimes some of these algorithms, if you read carefully their GitHub and if they are well organized, they actually give you guidelines. Like Banksy gives you guidelines. Like if it's, I think if you want domains, they advise 0.8. And when I tried, so I tried 0.2, 0.4 up to 1. And it's true, actually 0.8 worked fine, yeah, for me. And then for more cell type labeling, they actually advise 0.2. If I'm not wrong, I can't remember whether it's SSAM or whether it's SPA GCN. They actually give you, like, I think there's a supplementary note or something in their GitHub which says how to choose the kernel width, right? So the kernel width has to be close to your average cell width in your tissue. So it's helpful when they give all these guidelines. It means that they have thought about the biology. Yeah, so start there. Yeah, and if they haven't given, then you don't have a choice. You have to vary the parameters yourself, which can be very computationally intense.
Great. One more. You mentioned spatial clustering is not always necessary. Is there a way of knowing beforehand if it's worth running?
So, like I mentioned, I think, it's actually not so straightforward. When you run your non-spatial Leiden clustering, right, you should be able to see the broad domains. You should be able to see the patterns already forming, because no matter how mixed up tissue is in terms of its cell state or cell types, right? Sometimes there is always a tendency for certain cell types and cell states to aggregate. And we know this from literature too, right? A lot of literature in the past have guided us. There's a wealth of information out there. Like when we embark on a project, of course, if you're doing spatial, it's because you have some sense, some information that there is a spatial domain. I think that is your first point. And then there's a lot of imaging data set out there. The thing is, when we do spatial, we think sometimes maybe we forget that spatial is not just spatial transcriptomics. There's a wealth of images out there, H&E. You have the what do you call it? All these, can't remember specific names. You have all these large data sets with H&E, IF. You can always go and look at how the tissue looks like. Most likely, if you see structures in these images, whether it's H&E or a simple 2-marker, 3-marker staining, you will be able to see those domains there. Yeah, they will be representative. But yes, it is it's not a direct question. You can only have a sense, and you will have to try and test it out, which is why I suggest try it out with the simplest method so that you know, if it is worth moving on in that direction.
Great, thank you. And with that, unless there are any last questions, I think we're good to move to the lab portion.
Let me just maybe. Yeah, I just wanted to show you this. I mean, I hope it will be helpful. Okay, so I've been talking about all of these methods, right? But so just to give you an idea, so this is Banksy, right? It's also easy to read. So this is how the matrix looks like. So this is what they do. Apart from forming this 3-layered matrix, they have done nothing. They have not done anything spectacularly unique. It's very simple in a sense that the first matrix is your gene expression. The middle matrix is your mean expression of all your cells surrounding the query cell. And then the third matrix is the gradient of gene expression. And all they did was apply PCA and Leiden clustering over these 3 matrices. And then how they compute, right? So this is why, okay, maybe I'm being a bit too by harping on materials and methods, but the reason why I like to look at this is because it helps to actually understand, like, intuitively how the method works. And if it makes sense to you, right? So the first thing is they set up the spatial kernel. And it makes sense because you need to understand what your neighborhood is, right? So each cell has got n number of neighbors, right? Now, the thing is, you can do it by graph or you can do it by radii. There is no such thing, I believe, that a graph nearest neighborhood or k nearest neighborhood is more effective than a radius-based k nearest neighbor. It really depends. Sometimes there are cases that in organoids, right? You might just want to identify this neighborhood structure in a radius. And then it really does make sense in how they weighted it as well, because that neighborhood gene expression is important, right? It definitely is going to be different depending on the site and also the cell. And then the AGF was the one that really impressed me, because I've been always looking. So this is like my experience. I've always been looking for something that looks at gradients of gene expression, because that is something that's very important in GBM for me. So that was how my choice was motivated. And then the next thing is they really do have a very clean and clearly annotated GitHub repo. and everything is well organized. So one of the things that I want to highlight, right? In module 1, you learned about normalization. Now, all of these spatial algorithms, they use default normalization. They don't, I think there might be some papers that benchmark on different normalization, I cannot remember, but how you normalize, how you transform your data will matter, right? Because it will affect your neighborhood average gene expression and your gradient gene expression, right? So they cannot fine tune the first part, all right, but just know that they are taking in the process data from the first part, your normalization, your transformation. So all that matters, how much you filter and how much you don't filter actually matters, all right? And they make it clear here. And then what is important is this here, the lambda and the k-geom. So the lambda is basically telling the Gabor filter, how much should I look for the domain? How big a domain should I look for? All right. And this is a fine balance. You might think that, oh, so if that's the case, if it's a tissue microarray, I can use 0.2. It actually doesn't work that way, right? Because intuitively, when you give it a smaller domain, I'm not saying that it's really thinking, but maybe it is, I don't know. So how it's thinking is that it's thinking that smaller domain means that I am finding local patterns, which means that perhaps it's going to give you still the cell type rather than small structures. All right, because the domain is still small, the width is still small. And then another thing that really makes a difference is the k-geom, which is basically the number of neighbors that you want to compute. We are using 15 in this workshop, but that is only because if not, all your, VMs will crash, all right? But ideally, I use 50. You might want to use 100, depending, all right, on, again, the organizational value of your tissue as well. If you're looking at organoids, I wouldn't do 100 or 50, maybe 15 would be fine as well, or even 9 would be suitable. All right, so these are things, and they have given all these like parameter fine tuning, and it's there, so it's good. Now, I like SPA GCN, and I've used it. It didn't give me better results than Banksy, but it was easy to use, and conceptually, I liked how it was, how they represented it. So for SPA GCN, they take into account your HNE. Now, how they use the HNE image is another debate altogether, all right? So an image, right, in this case, they have used the gene expression, all right? And they have used the XY coordinates of your cells, all right, or your spots in this case. But the third dimension, so they call it the third dimension, not the third matrix. The third dimension comes from the image itself. So how they actually compute the third dimension is that they take a window, all right, and they move across the HNE image, and then they average out the RGB values. And then they do some sort of cleaning up, scaling of the RGB values. And then what the principle behind this, right, is the fact that if I have gene expression of my cells, all right, or all my cells have similar gene expression, but their RGB profile is different, it means that I am looking at some sort of dissimilarity as compared to a region where my RGB values and my gene expression values are concordant with each other. It makes sense, except that the thing that I question, and that's because I do image analysis, is that the value of organization from an image is not from RGB values. It's from the pixel organizational values, how the pixels are distributed. So I would argue against it. But in a way, but my point is, I could be wrong, right? So in principle, I don't agree with this. But the concept is there. The idea is there, and the idea is correct, right? That sometimes what you see in gene expression might not be the actual total truth. So they have integrated image. So the reason I is bringing this up, nothing, I didn't do anything. No, not me. Oh, okay. It wasn't me. It was this thing here. Okay. All right. So the reason why I bring this up, and I forgot what I wanted to say. Just give me 1 second. Okay, so the reason why I wanted to bring this up, forgot what I wanted to say. What was I saying before this? Yeah, something about image, right? Yeah, so it is, okay, it will come back after a while. Yeah. Okay, so the concept is that your image, right? So we are naive to think. So the thing is, when you are looking at an image, especially if it's an H&E image, right? You're not looking at, it's just RGB values. These are not fluorescents. So the overall RGB, what do you call it, distributional pattern is actually a net result of all your cell types, right? So which means that a disorganized cell type structure by gene expression will give you a uniform RGB, what do you call it, patch, right? So it is something that we fail to maybe remember, but we can use it, right? Because we see it often when we do, especially in H&E, right? Why is it that there are so many papers that successfully interpret gene expression from H&E? Because there is that connection between order and disorder, right? So that's the principle of it. All right, and then also, again, this is Python. It works very well. Banksy also has got a Python code, but you have to download the repo and then you need to direct it. It's not straightforward, but it's easy to use. I'm going to skip Fictor, but I just want to talk about Fictor and SSAM. Fictor and SSAM, can I find a nice picture? Right. So Fictor and SSAM, these are all what do you call it? Segmentation-free methods, okay? So what do we mean by segmentation-free methods? Which means that they look at your gene counts, all right, your genes, your expression values, and there is like a moving kernel, and they smooth things out. So you actually can identify cells, but the cells are not real cells, they are pseudo-colored. All right, so it will define. So if it's moving through a region that is acellular, it will just not map it. If it's moving through a region where there's cell and there's some sort of gene expression, it will map it up. All right, and the reason why I like this method, because I think this is something that you might want to try for Visium HT. I don't know how well it will work for a 2 micron or a 16 micron. According to, I think it was the SSAM paper, I can't remember, they actually have mentioned that the scale does not matter. You can have a large scale and it should be able to smooth things out because it's predictive, right? It's statistics. So it will statistically compute as it moves and it averages out using the kernel, and it will plot out what are the peaks of gene expression and where your cell centroids are. So I think this is something that's worth exploring, even though we won't be doing it for the workshop.